into the topic of today. So what I'll be talking about today, they are parasites. Are they friends? Are they foe? And how do we understand this relationship between the parasite and the host? The host could be us as humans. It could be, as I believe, our clients or our pets, such as the veterinary animals. And thank you for the introduction again, XWOW. That was a very good one. So here is the outline of what I'll be talking about today. I will give an overview of an introduction to the concept of parasitism, determinants of parasitism, which is also another term for the epidemiology of parasitic disease, and the kind of relationship that exists between the host and the parasite. And this particular aspect is a broad concept. So what I've just done is to give examples of different parasites, such as ectoparasites, endoparasite, and hemoparasite that affects animals, and just give a broad understanding of what kind of host immune host response are presented against them, and how these parasites, because they are very smart, and that's why we're still battling with like 90–95% of them up to this day. So they're very smart because they also present challenges to the host immune system too. And the parasites of veterinary importance. And I think the very big concept of today, which is the One Health, is the role of veterinarians in the control of parasitic diseases. And also what will be covered under this section is also zoonotic diseases. And then I'll take questions if time permits me. So the concept of parasitism, basically, it's more of like an ecological terms whereby parasites and parasitism are introduced when you're trying to classify or talk about relationship between an organism that takes its living or makes its living off of another organism. And not just that the organism making off its living is just doing that, it also presents some other challenges, such as health challenges to the host. So these are ecological terms that define a way of life rather than a coherent and evolutionary related group of organisms. So who or what is a parasite? What do we refer to as parasite? So these are organisms that derives nutritional benefit from the host and is usually considered harmful to the host. So I will just give you an idea of, just to have a concept or an understanding of the abundance of different organisms that could be parasitic to either us as humans or as animals. So if you look at this chart, right here is the vertebrates where humans and animals exist. And we can see we just make about 5% of the whole organism or biomass on the planet. And you can see like almost 70–75% of the chart is made up of invertebrates. And this invertebrate is this group of organisms that we find are parasites. Name them ectoparasites, endoparasites, hemoparasites. So you can see there's a lot of competition or a lot of struggle for the very small resources, which is we humans or animals. So these invertebrates, they compete amongst each other for the small resources that exist for their survival, because they need to survive, they need to procreate, they need to maintain their progeny, and they also need to evolve. So it's a challenge to them too, if you look at it from this angle. So what exactly is parasitism classified under? It's not just parasitism that exists. So parasitism, just like mutualism and commensalism, they are a form of symbiotic relationships. And what actually defines them is the tilt of the balance. Where exactly does the balance tilt to? In parasitism, the balance tilts towards more of the parasite, which is making a living off the host. Whereby in commensalism, as you can see, I mean, we all know about the ruminants, animals such as our cattle, our sheep and goats, where they have commensal bacteria in their GIT, in their rumen diet, them to break down the cellulose components of what they eat. So they have commensal bacteria that reside in their ruminal flora that produce enzymes that breaks down cellulose. So that's a form of symbiotic relationship whereby the organism benefits and also the host, which is the cat, to also benefit from ease of digestion of the cellulose. And mutualism also is another form of relationship whereby, and this is seen more in when you it's seen more in more larger organisms, not really in parasitic, not really in smaller to higher organisms, like just in the cattle-egrets and the cattle, where the cattle-egret can be seen picking off ectoparasites such as ticks and lice on the back of the cattle. And the cattle also provide a form of movement from one place to the other, with transport from one place to the other. And parasitism, as I've discussed earlier, and in this case, you can see a tick that is like very strongly attached to the skin of this particular animal, and the tick is going to be there for as long as it takes enough blood, and when it takes enough blood, it drops off. So this should give us an understanding of the kind of relationship that exists between organisms. And the point of note I will try to bring here is that the concept of this symbiotic relationship, especially parasitism, it's subject to the point in time whereby you examine or you look at that relationship. So a parasitic relationship could eventually switch to mutualism or commensalism, and also vice versa. So we have situations whereby a bacteria or a parasite is living in harmony within the host, and probably it's just exposed. There's just exposure to some harmful substance in the microenvironment of the host, and this sort of changes the genetic repertoire of the parasite, and they just evolve virulence traits and they harm the host. So if we all know the answer to that, then we'll all be free of the parasites that are challenging us in the world right now. So there's a borderline, there's a thin line between the existence of parasitism and the other forms of symbiotic relationship. So what I would like you to take away from this is it's a thin line. Parasitism can switch to mutualism and vice versa as well. So when we talk about parasitic disease or parasitism, there is when we look at from the epidemiological aspect, what used to exist is the hosts, which is the larger animal or the human, whereby the parasite feeds on, the parasite, which is the culprit, as well as the environment where these two exist in that form of relationship. Now, right now, there's a new player, which is the vector, and this is this area is a very critical aspect of research, because it just sort of amazes you as a scientist or as a veterinarian, because so what it means is the parasite can survive within the vector, which is oftentimes invertebrates like insects, and also survive within the host, which are vertebrates. And these two organisms, they present different challenges to this parasite. So it's a sort of you can see the parasite trying to maintain evolution, trying to survive by surviving in different forms of hosts. So if we can understand how the vector supports the growth of the parasite, then maybe we can break this cycle of the vector transmitting the parasites to us. And that's why I see a lot of research areas now where you see vector biologists doing researches in mosquito, ticks, sandflies, tsetsefly, so that we can understand what exactly is facilitating the transmission of those parasites from the vector to the host. So when we talk about the host, a lot of things comes into play that facilitates the establishment of parasitic infection within me or within my animals, within the cattle, within the sheep, within the goats or dogs. So the host basically should live in a very comfortable environment, should have a functional system such as the immune system, the defense system that prevents foreign organisms that are detrimental to its survival to come in. But there are other conditions that reduces the ability of the host to do those functions. And one of it is this. So what they do is they make the host susceptible to parasitic infections. And what are those things that can make the host susceptible is the fact that when the host is nursing an ongoing infection, or when they have a concurrent infection, because it's already battling on the war front with, it could be a bacteria, it could be a virus, or it could even be a protozoan or whatever, or it could be some malnutrition or some nutritional deficiency that is already making the host, that makes the host immune response weaken to defend itself against a secondary infection. So most times you see parasitic infection as concurrent infections or as secondary infections to an already established conditions, which made the host susceptible to new parasite infections. And there are also instances whereby the host is already used to a particular strain or a particular species of a parasite. So when there's an introduction of a new species or a new strain, most times it's always a new strain. So it's still the species, but because of some difference in the genetic combination, it presents a very new challenge to the host. So in that case, the host will need to go back to the scratch to start mounting the immune response. And that takes a long period of time. So between that window, the parasite already established itself and it causes infection. And there are instances also whereby there's absence of acquired immunity in the host, especially in young animals where they've not been exposed previously to a particular form of parasite, or when you move a group of animal, a group of flock of animal from one environment to the other. So because they don't have that immune memory that tells them, oh, there's this existing parasite here, they don't know how to mount an immune response to it. So it takes a while, and during that time, they become susceptible. And also, I've mentioned the strain of the parasite that is coming in. Some strain of parasite, they are just not pathogenic, which means they come in, they don't stimulate, they don't mount an immune response, whereby other strains comes in and they mount immune response to the host. And also genetic factors, and this is a very complex area where you see some strain of some species or some types of hosts, such as where we've seen in trypanotolerant cattle, sheep that are resistant to ticks infection. So some genetic factors comes to play there, whereby some breed or some species of hosts, they are resistant to parasite infection. And when it comes to the parasite, because the parasite also needs to, even if the host immune system is compromised, the parasite also needs to be able to survive within the host or maybe outside of the environment, because it's not all the time they will find a suitable host to live in. So there's going to be a window of time whereby they are lying outside of the environment within where the host is found. So it favors the parasite if it can survive for a longer period within the environment, within the immediate environment of the host. So especially this is more common when you have seasonal parasitism, whereby during spring or rainy season, there's a very high incidence of a particular parasitic disease because, during summer or dry season, there's a very high incidence of parasitic disease because animals converge together, maybe at waterfronts to take water, or they converge together at just small area to feed. So because of the availability of the host, the parasite has a lot of opportunity to infest either young or old or immunocompromised hosts. But in other seasons where animals are widely dispersed, there's less overpopulation in terms, overcrowding in terms from the aspect of the host. So it will do a lot of good for the parasite to maintain their presence in the environment. So they go into a form of dormancy and they can survive in a very long time. Especially they go into a form of dormancy where they have some form of protective coat or a protective covering that makes them survive throughout those window whereby they wait for the subsequent season to come. And also when they are inside the parasite as well, when they eventually enter into the host, because the host is not just lying down and waiting for them to come in, they need to mount a very strong immune response, and in which the parasite also respond. They evade immune responses of the host, such as in trypanosome, where they present different forms of surface glycoproteins that keeps confusing the host all the time when it comes to antibody production. So they evade host or they hide in places where the host immune response cannot even get to. And some are so very cunning in that they sort of, I don't know how to even put it, they sort of have this key hypothetically, they turn off some system in the host, and those system indirectly protects the parasite against the immune defense of the host. So it's very interesting relationship that occurs between the parasite and the host as well. And when it comes to the environment, because the environment is the sort of the is the it's just like into this to where these two organisms they meet and they converge and they interact. So in situations where an environment has been free of a particular parasite or a particular parasitic life stage, maybe you take your animal off a grazing pasture and you starve the pasture of animals feeding there. Probably you do a rotation system. So whatever life stage of the parasite is there, over time they're going to die. They're going to, because they can't find a host to maintain or survive. So when you bring another infected animal to the same environment or to the same pasture, they shed the intermediate life stage of those parasites, and the parasite also repopulates and they survive in the environment again. So bringing in infected stocks to a otherwise parasite-free environment will establish another parasitic infection in that environment. And also, this is more common in stock hard animals like cattle, sheep, and goats, where they are effluent or manures. They have a lot of nematode eggs, cysts, and even adult worms at times, whereby they can survive for longer period in the effluent. And what those effluent does, they serve as a form of transport, where they move eggs or life stages of a parasite from one particular, from a contaminated area to an otherwise clean area. So this is also a problem with that, with maintenance of these parasites within the ecosystem or within the immediate environment of the host. And the vector is also very important, because what the vector does is it gives these parasites an opportunity to develop part of it. So most of the time, the sexual forms of the parasite, they develop within the vector, such as in Plasmodium, they develop in the mosquito, and also the Lyme disease causing bacteria, Borrelia burgdorferi, develops inside the black leg ticks known as Ixodes capillaris before being transmitted to the host. So the parasite also has the means of surviving within the vector as well. So now when it comes to the kind of relationship that exists between the host and the parasite, I've made this a bit simpler in the sense that it goes beyond just the location of the parasite, as you can see here, and also the types of hosts. But I believe this should make it more simpler, because when you talk about the parasite location, these are things that we can appreciate easily. You talk about ectoparasites such as ticks, lice, mites. You talk about fleas, and you talk about protozoan parasites, which are hemoparasites, such as the Babesia, trypanosome, and also, or even Thalaria, which causes a lot of economic loss in cattle production in the tropics, and also roundworms, which are endoparasite, but are found more in the GIT. So based on this location, this different parasitic class, based on their location, they're exposed to different forms of challenges that the immune response of the host present to them. You don't expect the kind of immune response mounted against a tick to be the same as that mounted against a protozoa, or to be the same mounted against roundworms. And also we have the different types of hosts, such as definitive host, the intermediate host, and the paratenic host. In the definitive host, these are the hosts whereby you find the mature stage of the parasite, such as the adult worms, inside them. And what they actually do basically is the mature stage of the parasite, because they cause either obstruction of luminal organs, such as the GIT lumen, or obstruction of the heart, such as seen in heartworm disease in dogs, Dirophilaria immitis, or they even penetrate into tissues. So they migrate from one tissue to the other. And in doing so, they stimulate immune responses in this host. So the presence of the adult life stage of the parasite is also detrimental in the definitive host. And the intermediate host is where you see the developmental stage of the parasite in the intermediate host. And this is a snail. And this is a typical player in some worms, some nematodes of cattle, such as the Dicrocilium dendriticum or schistosome, or even trematodes of fish as well. where they use snail as their intermediate life stage before being picked up by cattle or sheep or even humans that are accidental hosts in some instances. And likewise, we have another form of host, which are the parathenic host. So basically, parathenic host is a form of host where you also find the developmental stage of the parasite residing in them, but they don't, they are form of like, how do I put, they are form of like a transport host. So they allow, they also, in an ecological point of view, they make the intermediate life stage of of the parasite available to the host. So most of the time they don't affect the the parasite don't affect the parathenic host most of the time. But in either case, in some instances, they've been seen to also, because they penetrate the skin, they've been shown to cause some form of tissue laceration. And when they move from one form of the tissue to the other, there's also a form of immune responses. But these are not really, unless the parathenic host is a host that has this huge economic significance. So if it affects, if the parathenic host, they are hosts that has a huge economic significance, maybe it's an industry whereby they make a lot of money for people. So this could be a problem. So based on this kind of host-parasite relationship, it's a form of, it's dynamic, it's continuous, it's always ongoing. And basically the host is sort of like, at some point, I feel the host just comes to an understanding that, okay, I'm just gonna let you be. So it's sort of like a truce that comes up between the parasite and the host. And that's why sometimes you don't even see the clinical signs of a parasitic infection in the host unless you're doing some other form of diagnosis. And you see the presence of maybe worm eggs in the feces or adult worms in the feces of the dogs or the cattle. They are there, but they are not causing significant, obvious clinical signs. That's because the host, there's this form of understanding between the immune response and the presence of the parasite. So the parasite virulence also determines its survival within the host. This is, to me, this is more like, so when you say the parasite virulence determines its survival within the host, because the more powerful the parasite is in the host, then the more significant the kind of host immune response, the kind of immune response that the host will mount against the parasite. So it's in the favor of the parasite, or it's in the betterment of the parasite when you're in the host, just have a very low profile. If there's anything like that, have a low profile and just maintain your life and don't challenge the host that much. And I think that's what most parasites want to do. They don't want to affect the host in any way. And I think it's the other way around. The host just trying to not be tolerant, just want to mount immune responses against the parasite. And the parasite will also respond back. So the degree of host resistance to parasite invasion depend on the immune defenses. So if the immune defense of the host is strong enough, in the first place, the parasite will not have a way to penetrate. And when they even do, if the host immune resistance or the immune defense is strong enough, they will, you would have seen situations whereby they they arrest the development of the parasite, or they reduce the productive efficiency, which is also known as the fecundity of the parasites. So when you reduce the production of the parasite, then they cannot produce much and they cannot survive for a long period of time. So it's a very dynamic interaction between the host and the parasite. So now I'm going to be, because if I give instances of parasitic infection, they are just too much, like different types. So I'm going to be talking about host resistance to parasite under a broad stage. And the first one I'll be talking about is the species resistance to parasitic infection. And this is very typical. I think the first thing that comes to my mind is they are the Emeria genus, because they are more like host specific. So the Emeria species that affects poultry will not be found infecting horses, and even if they are present there, they will not cause clinical signs within the horse. So there's host specificity to different forms of Emeria species, and this could also be found in different forms of other nematodes or cystodes or trematodes as well, whereby you only found a particular species of a genus in specific hosts. So what mediates the specificity is still an ongoing area of research if it's mediated by the presence by the parasite or it's mediated by the host. And most likely it's just a form of recognition whereby the host recognizes. So in the case of the host recognizes a Emiria leucati as a parasite or as a danger which should be eliminated. Whereby if Emiria tenella is found within the host probably don't recognize it as foreign, so it doesn't mount an immune response against it. So there's a form of species resistance whereby different forms of parasites or different species of parasite have different hosts that they are specific to. Another form of resistance is the age resistance. And this is a very common phenomenon that is seen whereby newborn or very young from animals, especially under the age of 1, are highly susceptible to nematode infection because they've not. So with nematodes, you need to get to a point where you acquire resistance based on age. So as you get challenged with the nematode, it reduces the efficiency of the adult nematodes to establish infection in the animal. And even when they do, their reproductive ability, the ability of them to successfully shed new eggs is also reduced. So a young calf or a young calf that is just giving birth to and is exposed to a pasture whereby it acquires the eggs or the worms of a typical nematode will come down with obvious clinical signs compared to an adult cattle or an adult cow. So there's age resistance, and this is a typical example that comes to my mind is the Strongyloides genus, whereby the young animals are highly susceptible, but as they grow older, you barely see clinical signs of diseases, but they still keep shedding the eggs in their feces. And the next one that I'll talk about is the breed resistance. And this is still an area of ongoing research to understand why some breed of animals are resistant to a form of parasitic infection that are otherwise known to not just cause infection in other breed, but also have been shown to be very, very detrimental to the survival of other breeds of animals. And a typical example are the trypanotolerant breed of cattle, such as the Indama cattle in West Africa, that have been shown to be resistant to be trypanotolerant. So, because trypanosome is a very smart parasite that confuses the host immune response, and over time they cause a chronic infection. We just keep causing a wasting disease, and the animal dies eventually. But in this breed of cattle, they've been shown to be resistant to trypanosome, and research is still ongoing to try and understand why it is so. And another form of resistance to a nematode, Imoncus contortus, is the red Maasai red Maasai goat that are found in South Africa. They've been shown to be highly resistant to the Baba poleworm, which is a Imoncus contortus, which is a GIT nematode. So this is also a form of breed resistance, and also the Maasai sheep in Australia have been shown to be less. They are not completely resistant. They are less susceptible to Strongyloides. So now I've broken the different kind of parasite that I could come up with into helminths, protozoa, and even arthropods. So I'll be talking about host resistance to this under the concept of the immune responses to these different forms of parasite. So basically, when an helminth, either a nematode, cystode, or trematode, is introduced or infects or breaks the barrier to find itself within the host, what you see is, because these are multicellular organisms, they are not like bacteria whereby the eosinophil or the white blood cells can just be activated and engulf them. These are multicellular organisms, and what you see is a myriad of host immune responses coming in just to kill this parasite. And in extension, these responses are also detrimental to the host. So what happens is you see a lot of responses from the eosinophil, which release mediators. The eosinophils they come in, they degranulate. The neutrophils, they degranulate. Monocyte, they degranulate. And what it does is there's a feedback mechanism by the mast cell, which further increases the permeability of the peripheral circulation to further release more of this white blood cell. And these are non-specific, basically. And now another thing you see is the B cells and T cells. And most of the time, the kind of T cell activation is the T helper cells too, that are activated against helminths such as cestode, nematode, or trematode. And what they do is they produce interleukins such as interleukin 4 and 7 are mostly responsible when it comes to helminth infection. So this develops mast cell receptors where they bind to the T cells and the B cells. They become activated and they bind to the mast cell. And one very important thing about mast cell is they have a lot of active granules which they degranulate and release on the surface of the helminth. And when the mast cell degranulates and they release their granules and expose it to the surface of the helminth, it further facilitates the binding of all these other blood cells to all these white blood cells to act against the helminth. So the kind of immune response you see here, the kind of antibody immune response you see here is the IgE-mediated antibody, which is mostly seen in helminth infection. But the IgE is only exposed to binding with eosinophil. The other binding of neutrophils and monocytes are non-specific polyclonal antibodies, but this is specific to helminth and it's only bind to eosinophils. Another thing you see is also complement activation. And complement activation further leads to activation of degranulation of mast cells to also degranulate and release vasoactive amines that also further stimulates antibody production. And when you see all of this going in, and what comes to your mind is, okay, this shouldn't even survive, but it will come as a surprise to you that this parasite, they found a way to evade this host immune response, as I said earlier, or to even hide themselves within some of these blood cells and some of these white blood cells or mast cells. And what that means is they don't get exposed to the mediators of immune response. So when you have acquired immunity or when you have immune response against this parasite, the success of the acquired immunity is highlighted by prevention of reinfection. So you want to expel, and you also want to expel this parasite. When you prevent reinfection, because they have the B cells, which forms memory cells at the end of the immune challenge, they are already exposed to that parasite. So they know what the parasite comes with. So when there's reinfection, the immune response in secondary infection is faster and more effective. And they also reduce the fecundity or the reproductive efficiency of this parasite. And finally, they facilitate parasite expulsion from within the host. So, but in other way, the parasite is not just going to sit there because the parasite wants to live, wants to survive, it wants to establish itself. So what the parasite does is, instead of this specific IgE being produced, the parasites stimulate the host to produce non-specific antibody. So those are polyclonal IgG. And once that happens, there is non-specificity of this IgG. The mast cell will not be able to bind. The eosinophil will not be able to bind. So the host is producing immune response, but they are not effective. They are not effective against this specific helminth. So the helminth stays longer within the host. And also, they also try to undergo concomitant infection, whereby they rapidly multiply within the host, they rapidly reproduce within the host, thereby overwhelming the host with their numbers. And every time the host needs to produce another effectors of immune response. And what that leads to is, it leads to a form of energy, whereby the immune cells are tired, they are weak, and even when they are producing effectors, they are not effective. They are not effective to remove the parasite. And finally, they also have some immunomodulative properties, whereby they also produce, because they are multicellular organisms, they produce proteins, they produce enzymes that binds or inhibits all these white blood cells. They inhibit the T helper cells, the B cells. And what that does is, when the soldiers are incapacitated, then they can't control the helminth. Now, another form of host resistance is to protozoan infection. This is also a bit similar to the helminth infection, but in a more different way. And you see a lot of cells coming in. We have the dendritic cells, the macrophages. The macrophages are important here because the macrophages, they engulf the trypanosome. And when they engulf, I mean, I just use trypanosome, it could be Leishmania, it could be Babesia, it could be Theileria. So they engulf the protozoan and they phagocytose and they release the lysosome or lysozymes, which degrade the parasite. You also have the CD8 T cells, the B cells, which produce antibody, the phagocytic cells, as well as complement activation. So in the presence of an effective immune response against a protozoan, what you see is the toll-like receptors immunoactivation, which produces toll-like receptor type 2, which stimulates the so what the toll-like receptor does is it stimulates further immunoactivation by the macrophage. It binds to the surface of the macrophages, and the macrophages acts as they act like antigen presenting cells. Yeah, they're able to identify the trypanosome and present them to the CD8 T cells and the B cells for antibody production. So the dendritic cells and the macrophages, they identify the surface protein on the protozoan, and they phagocytose and they present them to more specific immune cells that can produce specific antibodies. So the CD8 T cells and the B cells are involved in the antibody production that are specific to whatever protozoan is being presented. And also, you also have some other form of phagocytic cells that phagocytose or engulf the protozoan, and they produce a lysozyme that destroys the parasite. And finally, there's also complement activation, just as we have in the helminth infection. But what the parasite does, because they are smart, is they counterst every aspect of this production. They counterst them. So because we have different forms of TLRs. Some TLRs are involved in immune activation to counter the parasite, while some TLRs gives a negative feedback to the host immune cells. So the parasites stimulate the production of TLRs that are involved in negative feedback immune responses to the parasite. So in that case, the parasite can survive longer and stay longer within the host. And another thing that the protozoan also does is they exploit ROS, which is an acronym for reactive oxygen species, for their production. So within the macrophages, what they do is the cytosol of the macrophage is it's a very highly reactive environment that stimulates the production of reactive oxygen species. And what the reactive oxygen species does, it delyses the membrane of the trypanosome or the protozoan. So what the protozoan, because also the ROS is also detrimental to the host immune cells or to the host cells generally. And in production of the ROS, the host also produce antioxidants that protect themselves. So what the trip does is it hides behind the production of the host antioxidants. So the host is just producing ROS, reactive oxygen species, that is not having any effects on the trypanosome or on the protozoan. So they stay longer within the macrophage, but the macrophages are not effective in clearing them or presenting them to the B cells. So they also have some other surface proteins that bind to the receptors found on the CD8 T cells. And when they do so, they antagonize the effects of the CD8 T cells, thereby delaying the immune response. And the other thing they do that is similar to what the helminth does is they stimulate, the B cells are activated, right? But they stimulate the production of polyclonal and non-specific antibody. So the antibodies are just produced, but they are not specific. For instance, let's say it's Trypanosoma brucei. It's not specific to Trypanosoma brucei. It's just a non-specific antibody which delays the immune clearing and also gives the trypanosome a longer time to establish itself. So all of these are all marks of protozoan infection, especially trypanosome, because when all these things keeps going on, the immune cell still feels like we are not doing enough. They keep producing other mediators and other effectors. So the point where there is energy of the immune cells, and they become weakened, and they can no longer respond to the presence of the protozoan, and that leads to chronicity of the protozoan infection. And another thing that they do is the protozoan has a way of invading these phagocytic cells and stay within the phagolysosome. So the phagolysosome is when the phagocytic cells, when they engulf the protozoan, they have some organelles that contain degrading enzyme known as the lysosome. That's lysozyme that is meant to break down the cell wall and other parts of the protozoan. But the protozoan find a way to survive within the lysosome, which is very, very smart, if I'll say. So when they survive within the lysosome cannot degrade them, and they stay longer within the phagocytic cells. And all they do is just, when they see that there is a reduction in the immune response, they come out again and infect a new cell, thereby populating themselves. And they also inactivate complements and reduce the complement activation process against themselves. So all of this makes them more persistent in the host, and they can stay longer within the host, even in the presence of an effective immune response. So you could see a situation whereby the protozoan is infecting a very healthy animal, but over time the immune response of the animal becomes weakened, and it becomes even susceptible to other forms of infection along the line. And the other type of infection I'll be talking about are those initiated by arthropods, such as ticks, sandfly, kissing bugs, tsetse lice, and fleas. These are very fascinating to me, especially ticks, because I work with ticks, and so that's why I put the tick picture here. It's a very fascinating scenario because compared to every other ectoparasite that are known, ticks are known to feed for a very long time on the host. The average time that a tick will get attached to your body or to the body of the host is within 5 to 6 days. That's the least time a tick will spend on the body of the host. So the tick is on my body, or on the body of the dog, or on the body of the cattle, and the cattle doesn't even know the tick is there. It's taking blood meal. And it's fascinating that by the time they finish taking the blood, they could be 100 times their initial size. So they take a lot of blood. And it's not until I got to graduate school that when I started working with ticks, that I realized that I now understand, because when I see dogs and cattle that have ticks on their body, like hundreds of ticks, and they don't even bother to remove them, I just like, why would they allow them to stay that long on their body? And the ticks leaves when it wants to leave. It's not like the dog or the cattle pulls it off and removes it. So this is also a fascinating scenario whereby the ticks, when the ticks attach, or whenever mosquito, sandfly attach, they secrete their saliva, and the saliva is just like a micro pharmacy that has a lot of active components, proteins, enzymes, antibody antagonists that suppress every known form of immune response the host can ever think of mounting. So that is very fascinating. So to arthropod infection, to presence of ectoparasite or arthropod on the skin of the host, these are the immune cells that are mounted. Macrophage, dendritic cells, mast cells, and even the skin cell keratinocyte have been shown to be involved in immune signaling, although non-specific immune responses. And also the neutrophil, eosinophil, basophil, T helper cells, CD8 cell, B cells, and monocytes, they are all involved in fighting the presence of the tick on the body or on the host. So as I said, they feed for a very long time. The adult ticks can stay within 9 to 14 days on the host. And what makes them so fascinating is they have in the saliva, they have a lot of, as I said, a lot of molecules that they produce. And they also have, like the saliva has some analgesic properties, which inhibit the pain perception of the host. Unlike the sandfly, unlike tsetse, unlike mosquito, when a mosquito bites on you, you know there's a mosquito on your skin. You slap your skin to kill it or to ward it off. But the tick will be on your skin. You would never know it's there because they inhibit the pain perception, which is initiated by the bradykinin pathway in the host. So they can stay for as long as they want to on the host skin. But what is interesting is that repeated exposure by ticks or sandfly or fly has been shown to lead to immune development. And this is also seen in sheep and blowfly larvae, where when a sheep has been exposed to the larvae of a blowfly of the blowfly, they develop they develop a rapid immune response on secondary exposure to the blowfly. So that has also been shown on ticks and sandfly, whereby previous exposure on rabbits or dogs reduces the chance of susceptible infection as well. So the immune responses on subsequent infection, while effective, could also be detrimental to the host. This is very significant. This is important because the second time the tick or lice or mites are coming in, the host is ready. It already recognizes everything within the saliva. So it produces an aggressive immune response, which could also have a detrimental effect on the cells of the host, because they are most times non-specific and specific. So in that case, they also, while trying to reduce the attachment of the ectoparasite to the skin, they also expose their cells, or the host cell, to some damage of self-damage from the immune responses. So how does the saliva of the tick or ectoparasite counters everything that I've shown right there, which on a normal day should be able to ward off this invading ectoparasite? So they have a lot of research has shown a lot of proteins, a lot of enzymes such as SAP15, and this is known to inhibit the keratinocytes, and also they've been shown to even facilitate some pathogens from coming from the tick to the host. And also they have a lot, such as the metalloproteases. These are enzymes that breaks down the neutrophils or the white blood cells. And also some of these also inhibit complement activation. And some of these also prevent mast cell degranulation. And we all know when mast cell is not degranulating, or when the keratinocytes are not responding, or when the neutrophils are being destroyed, then nothing is there to identify this thing as foreign anymore. So they have a feed day to take as much blood as possible until they are satisfied. So as I said, the saliva of arthropod is just like a micropharmacy. Thousands of proteins have been identified from the saliva of ticks, mosquitoes, sandfly, fly, with pharmacological importance. And some of them has even been shown to also have some medicinal properties, such as anticoagulant that has been applied in surgery, or analgesic as well. And they consist of active substances that counters the immune mediators produced by the host. So to the final aspect of the talk. So what role do veterinarians play in the control of parasitic or zoonotic diseases? According to the American Veterinary Medical Association, One Health can be defined as a collaborative effort of multiple disciplines that work locally, nationally, and globally to attain optimal health for people, animals, and our environment. So this is a very big statement, and because we are not just thinking about, it's not an individualistic concept, it's the whole globe as a whole, the animals that lives in, the people that lives in, and even the environment that lives in. So there's a lot of disciplines coming into play to achieve this goal of One Health. So we veterinarians also have a role to play when it comes to One Health, abiding by the tenets of the One Health. And why that is important in our case is because majority of the emerging or re-emerging diseases that are coming in right now, like 70% of them are either parasitic or zoonotic diseases. And we are at the forefront of facing these parasitic diseases, or even they are either parasitic, vector-borne, or zoonotic diseases. And those three statements affect us more than any other profession. So we have a very big role to play when it comes to tackling the issue of parasitic and zoonotic disease outbreak, either emerging or re-emerging. So because we deal with these animals, we understand their behavior, we understand the epidemiology or the determinants of their existence. And even we have wildlife vets that deals with wild animals, which oftentimes serve as reservoir of some of these parasitic diseases or even viral diseases. And what happens is they amplify those viruses or those parasites in the wild. And when there is a spillover between, or when there's an interaction between the wild animal and the people, then there's spillover of those those viruses or parasites into the animal population, and that has been seen with recent occurrences of Ebola virus and the SARS-CoV-19 outbreak right now. So as veterinarians, we have, because all of these things are what we do on our daily, either as practicing vets, or you're involved in research, or you're even involved in teaching, or you're still a student. Diagnosis, surveillance, epidemiology, control, prevention, and elimination. These are things that have been postulated by the One Health Initiative that are important in identifying and reducing the occurrence of zoonotic diseases in the world currently. So as veterinarians, we have the skill to identify or to diagnose some of these diseases, such as even rabies, which are viral diseases. We do a lot of surveillance because we come across almost all of these animals on a day-to-day occurrence. And when it comes to the epidemiology of parasitic or zoonotic diseases, I feel veterinarians are more are at the forefront of understanding how what factors facilitate the establishment of parasitic infection, and in extension diseases generally within human and animal population, and control, prevention, and elimination. There are things that we are vast in that we've been used to, because we deal mostly with animals that live in a group, in an environment, and so we are facing that continuous challenge of coming up with approaches to reduce outbreak of diseases within even the small animal population that we manage or that we come across every day. So we have the epidemiological approaches and surveillance in order to inform control and prevention and elimination programs, so that we can have total elimination or reduction of zoonotic diseases in the world generally. So in continuation, just to emphasize why veterinarians are important in control parasitic and zoonotic diseases. So imagine and reimagine infections, they consist of 70% are vector-borne or zoonotic disease. And when I mean vectors like mosquitoes, the vectors chikungunya virus, West Nile virus, and vectors like ticks, the vectors Lyme disease, and vectors like sandfly, tsetse-fly, the vectors trypanosome. So a lot of them are there that are important because they help in amplifying majority of these viruses or these parasites in the human or animal population. And you can see the distribution of these vector-borne or zoonotic diseases in the world. And a typical example is the West Nile virus, which is the leading mosquito-borne disease in North America right now, and it's transmitted by this Culicoides mosquito. And we have humans and horses, which is not shown here as dead-end hosts. But we have horses and humans, they amplify these viruses. They are dead-end hosts. So, and also there are instances whereby there's human-to-human transmission when there's spillover into the human population. So it's important that, because these birds, which serve as the reservoir of this virus in the wild, these are what we work with. These are what we deal with. So understanding how these viruses affect these wild animals, or understanding how they amplify within the wild animal, and also because mosquitoes, they're also an area of our expertise as veterinary parasitologists as well, because we know their ecology, or we should know their ecology, we should know their behavior, we should know their biology, we should know how they can survive and transmit that virus to the humans or to the horses, which are also our patient too. So understanding the biology of the same thing with dengue fever, understanding the biology of these vectors, these mosquitoes, as well as the non-human primate, which could serve as dead-end hosts, or the bird, which could serve as the reservoir in the wild, is very important. And I think vets are more situated in this aspect to understand how to curtail the spread of parasitic or zoonotic diseases. And there are a lot more of them, like the Lyme disease, which is transmitted by which is transmitted by Ixodes scapularis ticks. And the reservoir is a mouse in North America here. We have the lymphatic filariasis, the leishmaniasis, Chagas disease, and even malaria. So what I believe, in my own opinion, is that because all these diseases, they have a single denominator, which is excuse me, which is the vector. They either use mosquito or ticks, or fly, or sandfly, or kissing bugs to transmit this parasite that they transmit. So we should, I think as veterinarians, we should go beyond just identifying clinical conditions, or because those are one-on-one event. Those are, you are dealing with an individual as a case. If you want to work under the tenets of the One Net initiative, you need to be able to think about diseases that has a broader implication, a broader effect, and that also has to play with amplification of these diseases in the environment, such as the role of these vectors that they play, such as ticks and mosquitoes. Why is it that just Ixodes scapularis can transmit this Lyme disease? Why is it that just the Culicoides can transmit this West Nile virus? And there are like 1,000 species of mosquitoes. There are 100 species of ticks. So when we start understanding how those vectors play a role in amplifying or in maintaining those viruses or those parasites or those bacteria within themselves before transmitting it to the animal or to the animal or humans, I think we are taking a step closer to not just to at least understanding how that happens, and maybe coming up with effective control measures to reduce the incidence of parasitic disease and in extension zoonotic diseases, which oftentimes start from the animal side of the One Health aspect. And just to emphasize furthermore, this is actually an old data anyway, but currently like 700,000 people die of vector-borne diseases every year in the world. And we could see the distribution of all these things in Africa, in South America. So these are areas whereby there's a lot of problem with vector control, with controlling of these ectoparasites such as mosquitoes, ticks, sandfly, tsetse. So there's a lot of problem with control of those vectors, and those vectors are important in amplifying those parasites or those viruses or those pathogens generally. And another thing is also the fact that we are not environmentally friendly in these areas. There's a lot of deforestation, improper land use. So when all those things keeps going on, we keep bringing ourselves closer to the wide amplifiers of these pathogens, and we keep exposing ourselves. And we also keep introducing ourselves to new vectors that are not otherwise known to affect us initially, just as we've seen in the recent Ebola outbreak. So just to summarize why I think the veterinarians are important in the control of parasitic or zoonotic diseases, it's been shown that veterinarians are more committed and better equipped to protecting animal health and welfare. When it comes to animal health and welfare, there's no other person you want to think of than to look for a veterinarian to take the issue of animal health and welfare. We take it seriously. This is what we dedicated our life right from the first year of veterinary school to the last year of veterinary school. And this is what we also swore an oath to protect the health of animals and even humans in extension. So this is very important because I think we need to start championing this cause, and we need to also take it as a responsibility, either by getting more information, getting more knowledge, trying to know our role in the whole concept of the One Health initiative, and trying to understand how we can play a role in our small areas and eventually in the big scheme of things. And also, in addition, our knowledge base is equipped when it comes to the diseases that can be transferred between people and animals. So even diseases that has not been transferred from animals to humans, even zoonotic diseases that has never happened, we have an understanding of those, how diseases can spill over from animals to humans and vice versa. Because we studied epidemiology in school, we studied medicine in school, we know how that happens, and we already know how to put those things in place to prevent the spillover of animal diseases into the human population. We also have a role in protecting human health as well, because we work in places like slaughterhouses to identify impending diseases that could be introduced through introduced into the human population via consumption of unwholesome food animals. And finally, our understanding in expert areas such as toxicology, pharmacology, and epidemiology are crucial to the issue of environmental health. Because when it comes to environmental health, it goes beyond knowing public health. It goes beyond knowing medicine. These are other contemporary areas where we've also been fortunate to touch on. We did, I mean, as a veterinary student, you're trained, you also take toxicology classes, you took epidemiology classes, and these in extension that aspect of sciences that deals with environmental health as a whole. So I believe we are well equipped to face the tenet, or to take the mantle of fighting the cause of One Health as a whole. So that'll be the end of my presentation. Thank you all for listening, and I will take comments and questions. Yes, yes. Yeah, it is. It's fascinating because even if a tick is on your body, you never know unless you're probably grooming your body and you see that lump on your skin. They're just so fascinating as an organism. I mean, I never liked them before, but when I started to have to like them, right? Oh, that would be cool. That would be cool.
Hello, good afternoon.
Hello, can you hear me? Hi, Abdul. Thanks for that very informative presentation. I am Carl, a veterinarian from the Philippines, and I am just concerned with the economic impacts if there is no intervention to eliminate, for example, vector-borne diseases because of the possible catastrophic effects as a result of endemic instability. For example, in cases of anaplasmosis, now treatment to eliminate anaplasma infection in most endemic areas is sometimes contraindicated because of a very high risk of reinfection and the relative susceptibility of the host population in the absence of any long-term immunity, which accompanies low-level persistent infections or immunity. Any thoughts about that?
Okay, so for the economic significance, so basically the vector-borne diseases, they are fascinating in a way whereby they amplify some of these pathogens. And what research is trying to do right now is to see if they can develop some form of technology to control, not just looking at the host right now, because when it comes to the host, you're talking of using antibiotics, you're talking of using vaccines, which are not most times economically significant or economically effective, even to the farmers that own these animals. So what research is trying to do right now is to see if we can interfere with the development of some of these parasites that we've mentioned, such as the protozoan, within the vectors themselves. And I'm sure, just not to bore you, but you would have heard of some technology such as the CRISPR, where they're trying to develop parasite resistance, mosquito, ticks, or even tsetse fly, so that they can be resistant to acquiring this pathogen from infected animals. So when they do that, so you sort of break the cycle of transmission from the parasite to from the vector to the host. The parasite cannot develop successfully within the vector, and where that does not happen, then they cannot transmit themselves to the host. But currently, we still go with the traditional way of treating, routine treatment, and over time, it's not always, it's what we do right now, but it's not cost effective, and it's not even economical, as you've said.
Okay, thank you.
You're welcome.
Hello. Yes, can you hear me?
Yes. Okay.
Yes. Hi, Abdulsalam. How are you, sir? Great
Yeah.
lecture, very informative lecture. In fact, I would like to ask question about cross immunity. The impact of cross immunity in helminth infection or protozoan infection. What is the impact of cross immunity in these sort of infections?
Okay.
Like
So, okay.
like between there are many certain helminths and protozoan being transferred from one species to another species, like there is a human primates, and the transfers of species from the other species like cattle and buffaloes, and sometimes there are some many species from one species to another species. Can you please demonstrate this, the impact of cross immunity?
So basically, if I the way I understand how that works is because most of the animals we deal with as veterinarians, they are herd animals like cattle, sheep, and goats. They live in an environment, and oftentimes you don't even see all of them coming down with with the infection at once. So so it happens whereby when you have a low dose of a parasite existing in in an animal, for instance, maybe a species of Homunculus, and because the host has been able to mount an infection against that, when another species of Homunculus comes in, there could be, I mean, this is, it's subjective the way I say it. It's a subjective phenomenon. It's not oftentimes what happens, but I think it's basically depend on the individual immune response. It's not what I think can be applied on a broader scheme of when it comes to parasitic infection. You see this more in viral infections whereby like different flaviviruses, you see hosts being exposed to one form of flavivirus develop strong resistance to another form of flavivirus or another strain of flavivirus. But when it comes to parasite, I think it's not something that is highly effective when cross immunity comes into play. It happens, but it's not effective in completely clearing the parasitic infection.
Thank you so much.
Yep. Yeah. Am I audible to you?
Yes.
Yeah. Okay. So I'm Dr. Muhammad Ridwan Pasha from Bangladesh. Good evening from here. I have I have one question. Is what do you think, Mr. Salam, that the whole world, every scientist are thinking about the microbial resistance in case of bacteria or other species, protozoa or something, but we are not thinking about the helminths. what do you think about the irresponsible use of the drugs in case of the helminthicidal drug, and what can happen after 20 or 40 years in terms of anti-parasitic drug resistance? That's my question.
That's a very good question. So with I mean, advocacy is going on all around when it comes to antimicrobial resistance, judicial use of drugs, and using using antibiotics responsibly when it's needed. I think the challenge with helminth infection is, one, it's a big challenge to come up with vaccines. So there's a restriction. I mean, you can count on top of your finger effective helminth vaccines that are available right now. I'm not sure they're up to 5. So, and with the parasitic infection, there's a lot of anti-helminthic drugs. And if used effectively, with my experience, with what I've seen, if used effectively, if used judiciously, they are effective. And one fascinating thing about helminths is because they have different forms, different life stages. So there are different forms of anti-helminthic that are effective against different life stages of the developmental stage of the helminths. But that said, with what we've seen in resistance coming up with bacteria, I think it will be responsible enough for us to come up with other approaches to counter helminth infection and not just depend solely on anti-helminthic, because we know what could happen. There could be resistance over time. So it will be a very good approach to start investigating other forms of control. And that's really why that's where right now there's more of an integrative approach to parasitic or helminth control, where you have an integrated system. You control the environment to reduce the parasite burden. You reduce the exposure of the hosts of the animals to the parasites, and you also sort of kind of eliminate or remove the vectors or the carriers of those parasites, such as in case of some helminths, such as snails, within the environment of your immediate animal. So basically, integrated approach of parasite control is the best way to go for it. We can't just depend solely on anti-helminthic, because if you look at the determinant, it's a triangle. You see a connection between the host, the parasite, and the environment. So if you can't just clip one connection and the other will not be there. So if you can interfere with all those other connections and reduce the abundance of the parasite or the helminth in the environment, you reduce the amount of the parasite present in the environment by reducing the carriers or the vectors. Then you also work on the host, give them a very good and convenient environment, boost their immune system. That's the approach to use, even in humans right now. That's the approach that's the integrated approach where a lot of professionals are coming in. And I think that's the tenets of the One Health initiative as well.
Thank you for a nice answer.
Okay.
Okay, good afternoon. Can you hear me?
Yeah.
Yeah. Okay, I'm Dr. Kauzar Kolawole from Nigeria. Are you there?
Yeah, go ahead, please.
Okay, actually my own question, and again, I want to actually thanks Dr. Salam for the insightful presentation. And my own question is just about the parasite and vector relationship. How does it actually work? Does the vector actually allows the parasite to come in and just establish in them, or the parasite has a way of actually trying to prevent them from establishing in them? Because if there's a way this vector actually have a way they can actually see how this parasite actually establish in them, I think that can actually help us in trying to put a kind of measure in the transmission, or maybe if you want to actually come up with a vaccine, because this is in the case of relation between anaplasma and tick, where the anaplasma actually established in the midgut of the tick before the transmission. So I'm trying. Does the parasite actually harm the tick, the vector itself, or they don't have any effect on the vector? That's my question. Thanks very much.
Thank you very much, Dr. Kolawale. That's a very, I mean, if researchers working on vector biology, if they can answer that question today, I'm sure that would be like a giant step for me. We won't go beyond us landing on moon because we don't see in the mind of the parasite, of the vector, excuse me, because now we know as humans or as animals that whenever anaplasma is in the cattle or whenever trypanosome is in the cattle, it causes so and so clinical signs. We know it's detrimental to survival. So what we don't know right now is, are these parasites actually pathogenic to the vector as well? That's one. If they are, what kind of immune responses is being put in place by the parasites by the vector to the parasite? And mind you, we have different evolutionary immune response when it comes to invertebrate and vertebrate. As humans, we have, or humans and animals, we have both humoral and innate immune response. In this vector, they only have innate immune response. They don't have humoral immune response. So it's a very complex scenario. But what I can tell you with what I've seen, with what I've reviewed in literature is when these parasites are in the vector, they come in as a challenge to the vector. They stimulate immune responses in the vector, telling you that they are not all coming in peace. They're also coming in to challenge the vector. So the vector mount and even in case of trypanosome and tsetse-fly, studies have shown that if you have like 100% of trips entering the tsetse-fly, what ends up getting to the salivary gland is less than 10% of the trypanosome that colonized the tsetse-fly in the first place. Just to let you know that the vector has a way of also modulating the amount of that present in itself at a particular point in. But so what scientists are trying to take advantage right now is to see if we can interfere with the development of those parasites within the vector, because they are the ones facilitating the vector. Malaria cannot enter my body without the mosquito present and biting me. Lyme disease cannot enter my body without the tick biting me. So if we can interfere with the development of those parasites, the vector, then we can, that would be a great step. And just to answer your question, they're also challenging vector too, because the vector are mounting immune responses. And they also interact with other forms of commensal microorganisms within the vector. Because just like us, the vector also has microbial organisms that benefit them nutritionally. So it's also a war of all against all with the parasite to survive.
All right, thanks very much, Dr. Abdulsalam Adegoke.
All right, thank you very much. So it seems we have a couple more questions. So do you have time, Abdulsalam?
Yes, I think I do have time.
Oh, that's great. So I'll just select the few before we wrap up. Let's see who else. Let's try unmute Richard. Nope, not working. Let's try somebody else.
Hello.
Oh, hi, Richard. Hi.
I'm good.
Yeah,
yes. Yes. Am I clear?
Yeah. Nice and clear.
Okay. I'm a student in Uganda, in Gulu University. I would like to understand from Dr. Abdulsalam about other intervention apart from the chemical intervention that we have overexploited from Uganda, like the use of deltamethrin, cypermethrin, intake control. You said you have worked with the ticks and you like working with ticks, and I don't know whether you have gone as far as exploiting the genetic potential of ticks so that they are easily controlled. Thank you.
So, I think I can split that question into two components. The first one is the control alternative to chemical control of ticks as a use of pesticide. So when it comes to tick, with tick research, it's sort of like it's still years behind other important vectors such as mosquitoes research. So with tick, the only available form of alternative to pesticide is a vaccine. I think it's called BM88 vaccine against Rhipicephalus microplastics that is commercially available for use. But right now, in the field in practice, I'm pretty sure it's insecticide that is being used. But when it comes to the genetic manipulation of ticks, there are a lot of labs here in the United States and other countries that have been trying to use the gene editing approach to make resistant ticks, such as being used in mosquito, to make resistant ticks so that they can be resistant to this bacteria or pathogen that they transmit. Pretty sure those are ongoing. But what I do in my lab that I'm here, another area of research that I work in, the area of research that I work in my lab here is to understand the microbial community. So this, just like in humans too, that we have a commensal bacteria within us on our skin, in our GIT, all these vectors also have their microbial communities within them too. So this, just like a question asked earlier, these microbial communities, they interact with the pathogens that it transmits to. So what research has been trying to say is if there's any form of antagonism, if there's any form of some antagonistic relationship between the microbial organisms within the ticks, as well as the pathogens that they are transmitting. So if that can be taken advantage of, that's what I'm trying to, that's the question I'm trying to ask and probably answer. Because it has been shown in mosquito, there's this particular species of bacteria known as Wolbachia, has been shown to interfere with acquisition and transmission of viruses like dengue viruses. So in the presence of Wolbachia, some mosquito species cannot acquire dengue viruses. And field trial of that has been tried in South American countries. So that's an area of research that is ongoing. But to answer your question, only one vaccine is available for tick, and there's even no cross protection. So it's just specific for Rhipicephalus microplus and not just for the ticks. But studies are also going to try and identify, just like I show you the slide where the saliva of the ticks has a lot of vasoactive, a lot of components. If those components have been investigated to see if they can be used as vaccine candidates to prevent the ticks from even attaching successfully, or to prevent them from blocking my pain perception, so the animal or the host can know there's a tick on my body and you can remove them before they transmit infection. And one fascinating thing about ticks is before they need to attach for 12 to 24 hours before they can start transmitting infection. So if we can detect them within that window of attachment, within less than a day, you can prevent them from transmitting whatever pathogen they have in their saliva to you.
Hello.
Yeah.
Hello.
Thank you.
Hello.
Thank you for the previous answer. So,
hello.
Yeah, we can hear you. So let's have two more questions before we close the event. So, yep, Dr. Mujab. Yep.
Yeah. Hello. Good evening. I am Dr. Mujib from India. First of all, I heartily congratulate Dr. Abdulsalam for a very nice and useful information.
Thank you, sir.
And my question is, what are the important some parasitic diseases, zoonotic diseases, which are very important for vets who are regularly in contact with domestic animals or pets?
Come again, please, sir. I didn't get that question clearly.
Hello, Dr. Mujab. Can you hear us? Oh.
I didn't get his question.
Yeah, I think we lost him. Shall we move on to the next question then? We'll come back to him when he can hear us again. So, let's try. Hello, Adegoke.
Hi, Ding.
Hi. No,
Hi.
I'm Panda.
Panda. Hi, Panda. All right. Hi, Abdulsalam.
Hi.
I'm Idoko from University of Abuja, Nigeria.
Nice.
I'm so glad to hear about your nice lectures today. Back home in Nigeria, we've done very little work on confirming vectorial capacity to transmit so many pathogens in animals. For example, I work with horses. I've detected several genotypes of Theileria that infect horses, but unfortunately, most of the ticks that have been reported in Nigeria since like the 70s, very little have been done to confirm their vectorial capacity to transmit this particular this pathogens of paroplasmosis in horses. Based on experience, I don't know whether you've done something on that before you traveled, or you're doing something related while in the US. But back home here, there is this problem because we have like 6 different species of ticks that are epidemiologically associated with pathogens of livestock in Nigeria, but confirming their capacity to transmit these pathogens have not been done. Which is very, very good to actually say, okay, I found this tick in this animal, and I can confirm that this tick have the so parasite in them, and they can transmit them they can transmit those pathogens to animals. I just want to share a bit, maybe some of your experience on what's on this particular area.
Okay, thank you, Dr. Idoko. So, so basically, I think that's a very good point, because just identifying pathogen within the vector doesn't translate to the vector being able to transmit it to a host successfully. So just to talk about vectorial capacity, in case for those that don't understand. So when we talk about vector capacity, is the ability of a particular vector, ticks, mosquito, tsetse to acquire a pathogen from an infected host and support the development of that pathogen and successfully transmit it to a susceptible host. So you need an infected host, a vector, and a susceptible host. So identifying a pathogen or a bacteria or a parasite within a vector and just streamlining and just screening up on that and saying, oh, this could transmit it, it's not enough. So basically what is done is to do this under experimental conditions, have laboratory animals, infect them artificially with the pathogen of choice, and feed them with clean ticks or clean vectors, check that they acquire that, and check that they can also successfully transmit it to another host that you presented. This is easier said than done because it's highly expensive for you to get the animals, for you to get the clean ticks or clean mosquito, and for you to also do a lot of ethical protocol, ethical consideration. I think that's why it's been a bit slow, because it's expensive to do such a research, but it's definitely going to add a lot of information to inform epidemiological decisions. So, I know there's a lot of detection of this using gene sequencing, DNA sequencing, PCR. That is not enough to validate that those particular pathogens can actually transmit those particular vector can actually transmit the pathogen. So the simple way to carry out that is to do what is known as a transmission study or transmission experiment, where you, under laboratory condition, you experimentally infect. In fact, you need to first identify the suitable host for the pathogen you're talking about. There are different laboratories. So the first one is even to identify what host can successfully be infected with the bacteria or the parasite, and also maintain it without dying, or without dying within the period of the time you want to do the experiment. After you identify that, then you can now go ahead and get your host, infect them, check that they are infected by maybe checking the blood, doing PCR, doing microscopic culturing them, and see that they're actually facilitating the pathogen development or the parasite development, and then put the vector that you have in mind on top of on them, infest them, and let them take the parasite from them. And then you can now you can now put them on the host that you are available, the clean host, and see if they can actually successfully transmit it to them. So in the nutshell, that's a very complicated and expensive procedure where I just hope we can get there in the not too far future, and because it will always help us to provide information on what we have within our country, and it will help us to inform decisions, such as you being an equine veterinarian. You'll be able to at least emphatically say this pathogen actually being transmitted by this tick, species of tick, and you have to be careful, you have to do this. I hope I answered that.
Oh, let's unmute Adegoke. Yep. He's not responding. Let's
Yeah, thank you very much, Abdulsalam. I think that's cool.
yeah, great. So I'll just try to unmute Dr. Mujib again, see if he can repeat his question. If not, let's end the session. So yep, it's not responding. Right. So yeah. So let's end the session then. So thank you so much. Thank you so much, Abdulsalam. There was an eye-opening lecture for me as well, and I'm so glad to have you to give the lecture today, and I'm sure everybody else like you so much. As you can see in our chat window, like we see people were just thank you for the great presentation, and they've learned so much. So
it's a pleasure.
Yeah, so as a tradition, so so let's I'm gonna allow everybody to unmute them. So as a tradition, we will have this everybody, if you can turn on your video and audio, and that's just say thank you, Abdulsalam.
Thanks.
Thank you, Abdulsalam.
Thank you, Abdulsalam.
Thank you very much, Dr. Abdulsalam.
Thank you very much.
Thank you, Abdulsalam.
We are so glad. Thank you so much.
Thank you very much for your
Thanks.
Thank you very much.
Thank you very much.
Thank you very much
so much.
Thank you very much.
Gracias, muy bien.
Very much for the presentation.
Yeah, thank you so much. Thank you, everyone.
Thank you very much. Thank you very much.
I hope you all enjoyed this lecture, and I hope Abdulsalam enjoyed giving us.
I did.
Yeah.
I did.
Yeah.
Thank you very much.
Cool. And let's end this session. I'm so sorry about at the beginning the internet issue, but I'm really glad you all stayed for this long, and I hope to see you next time. So I'm gonna just you're welcome to leave now, and I'm just gonna play
Thank you very much.
Okay. See you guys. Bye.
Bye, sir.
Bye, everybody. And see you.
Thank you, Panda. First of all, thank you, Panda.
Thank you so much. Nice having you. Bye.
Thank you so much.
Bye.
Thank you.
Miss you all. Bye.
Bye.
Thank you. Bye.
Bye.
Bye bye. Bye bye.
Bye.
Bye-bye.
Bye bye.
Bye bye. Bye bye. Bye.
Thank you.
Thank you.
Thank you.
Thank you. Thank you.
Thank you.
Bye-bye.
Bye bye.
See you again.
See you soon. Thank you very much.
Bye bye.
Bye bye.
Bye. All. See you all.